Hello, everyone, and welcome to today's webinar, a novel method for the isolation of highly pure carcinoma cell populations from FFPE tissue, improves sensitivity for detecting somatic mutations. I am Sabrina Lemus of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit www.miltenyibiotech.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located to the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Carina Emery, the Product Manager for Sample Preparation at Miltenyi Biotec. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Carina, welcome. You may now begin your presentation.
So today I'm going to talk to you guys a little bit about, we have a really great novel solution for using FFPE tissue for genomic analysis. Before I get too much into the details of that, let's just take a minute to talk about what is FFPE tissue. So FFPE stands for formalin fixed paraffin embedded, and it's a common method used to preserve tissue specimens for histological analysis. So the way this works is first the tissue specimen is fixed in formaldehyde. Next, it's dehydrated in a series of increasing concentrations of an organic solvent like ethanol. And then finally, it's embedded in a paraffin wax block. The block makes it easier to cut thin slices for mounting on a microscopic slide. This method of preservation enables biological specimens to be stored at room temperature for years and often decades. The minimal cost required to store FFPE tissue has resulted in the accumulation of vast archives of FFPE tissue blocks. It's been estimated that over 20 million FFPE blocks are archived each year in the US. So while FFPE tissue has traditionally been used for immunohistochemical studies, it's also a very valuable tissue source for genomic studies of both RNA and DNA. This allows investigators to correlate disease state and tissue morphology with genotype and gene expression signatures. However, working with FFPE tissue for genomic studies does come with some challenges. So before we move on, let's go ahead and set some expectations. So there are some technical limitations of working with FFPE tissue. The fixation process causes cross-linking of the DNA, the RNA, and the proteins, and this leads to fragmentation of the DNA and the RNA and denaturation of the epitopes. Moreover, there's really not a global standard protocol for preparing FFPE tissue and storing FFPE tissue. So the quality of RNA and DNA may vary significantly from one sample to the next. On the other hand, a variety of commercially available kits have been developed to standardize the molecular analysis of FFPE tissue and have come up with really clever ways to deal with the fact that the RNA and DNA from FFPE tissue is going to be degraded. And numerous studies have demonstrated the utility of FFPE tissue for genome-wide association studies and gene expression analysis. So even though there are challenges to working with FFPE tissue, it's such a valuable resource that we're coming up with more and more innovative ways to take advantage of this. So tumor is one of the most common types of tissue found in FFPE archives. but molecular analysis of tumor is complicated by tumor heterogeneity. In addition to having a diversity of cancer cells, tumors are also made up of the surrounding stromal cells, such as vascular endothelial cells, pericytes, fibroblasts, and tumor-infiltrating immune cells. Stromal cells make up the tumor microenvironment and are genetically distinct from the lineage of cancer cells. While cancer cells tend to accumulate mutations over time, the surrounding stromal cells do not, and they maintain a genome that is more comparable to germline DNA. Because of this, there are some issues with working with FFPE tissue. So the presence of non-tumor cells within a tumor complicates genomic analysis because cancer-associated mutation or gene expression signatures will be diluted out by the presence of normal DNA. One way labs address this complexity is to compare the FFPE tumor DNA to a normal matched sample of DNA. Novel mutations that are present in the tumor but not present in the matched normal tissue are considered to be associated with the cancer pathology. However, one of the problems is there's often no matched normal tissue available for comparison of tumor versus non-tumor DNA. The lack of a matched normal tissue is considered an obstacle to taking advantage of many FFPE tissue archives. So we're going to present our unique solution today to help overcome this challenge. But first, let's review what methods are currently used to deal with tumor heterogeneity in FFPE tissue. There are three common methods for enriching for tumor cells in FFPE tissue. Each method relies on staining the tissue sections followed by microscopic analysis. Typically, a trained pathologist will locate areas of the tumor and normal tissue. And as you can see in our first slide here, they'll often do this simply by just circling the tumor tissue on the slide. So one strategy for dealing with the fact that tumor samplers are going to be a mixture of normal and tumor tissue is to just simply accept the contamination. So you could just skip performing tumor cell enrichment. And by doing this, you typically are limited to working with samples to high to moderate percentage of tumor cells. Because when the percentage of tumor cells is really low, your ability to detect cancer-associated mutations is going to be compromised. The next option that is quite common is manual microdissection. So this is where, after the pathologist circles tumor tissues on a slide, you would then go in with the scalpel to selectively scrape off the tumor tissue. So macrodissection is effective at removing a good amount of the surrounding normal tissue. However, it cannot eliminate any stromal cells or immune cells that have infiltrated the tumor and that make up the tumor microenvironment. So this infiltration of non-tumor cells into a tumor can be highly variable. It can be minimal or it could be a lot. So the effectiveness of macrodissection is also going to be highly variable. So the last option is laser microdissection. So this is going to be a much more precise option. You can use it to isolate single cells or larger areas of interest under direct microscopic visualization. While laser microdissection is very precise and you can really selectively obtain pure tumor cell populations, it's very expensive equipment, and it's also quite time consuming and consequently low throughput. So Miltenyi Biotec has come up with an innovative solution that is faster and higher throughput than laser microdissection, but more effective than manual macrodissection. So it's kind of sits in between those two options. So let me introduce our method. Our novel method starts with the premise that the best way to separate cells into different populations is to first dissociate the tissue into a single cell suspension. Once the FFPE tissue is in a single cell suspension, protein markers can be used to separate out tumor cells from non-tumor cells, either by using flow sorting or MACS technology. Analyzing the tumor and non-tumor cells as separate populations not only enables increased sensitivity for analyzing the tumor mutational burden, but it also actually creates a matched normal DNA sample out of the same FFPE tissue block. So while dissociation of solid tissues into single-cell suspensions is commonplace for fresh tissue for applications such as flow cytometry and single-cell sequencing, this has not really been widely applied to FFPE tissue, and Miltenyi Biotec is trying to change that with the introduction of our FFPE Tissue Dissociation Kit. This kit uses gentleMACS technology to dissociate FFPE tissue to create suspensions of single cells, and it's able to do so while preserving important epitopes that can later be used for cell separation. Now let's take a closer look at how gentleMACS technology works. The gentleMACS system combines mechanical disruption and enzymatic digestion. Mechanical disruption is provided by the gentleMACS dissociation tubes. These tubes contain a rotor-stator system that gently breaks down the tissue. Enzymes are provided in a collection of tissue-specific dissociation kits. These enzymes are highly purified, pre-titrated, and they digest the extracellular matrix to yield intact individual cells. This is all controlled by the instrument and its collection of tissue and kit-specific programs. The instrument shown here is the gentleMACS Octo with heaters. By combining physical disruption with enzymatic digestion, gentleMACS technology allows for fast and gentle tissue dissociation. Because this can be done in a fully automated walk-away fashion, one can reduce hands-on time and decrease variability as a result of tissue processing. The FFPE dissociation kit is used with the gentleMACS Octo dissociator with heaters to process fixed tissue into single cells for downstream separation, DNA isolation, and analysis. This kit provides the protocol and all the reagents you need, starting from deparaffинization, going all the way through dissociation. This standardizes the dissociation process and avoids the use of manual methods that can be variable, time-consuming, and less effective. So by using this reproducible, standardized, and fully automated system, you can process 8 FFPE samples in parallel, and this will generate single-cell suspensions ready for whatever your downstream application is going to be: flow cytometry, flow sorting, cell separation, or DNA analysis. Miltenyi has validated this kit on a wide variety of FFPE tumor types, including breast, prostate, colon, lung, and pancreas. Now let's look at the workflow. The workflow starts with a 50-micron slice from a paraffin block. After slicing, the section rolls up on itself to create a paraffin curl, and up to 2 of these curls can be transferred into a gentleMACS tube. This method will be less effective with thinner sections because there will be fewer intact cells in thinner sections and no intact cells if the section is too thin. To prepare FFPE curls for dissociation, the paraffin is removed using the organic solvent xylene. And once the paraffin is removed, the tissue is rehydrated using multiple ethanol washes, followed by a final wash in distilled water. The sample is then taken through an antigen retrieval step, so it's heated at 80 degrees in a buffer, and this reverses the cross-linking that was introduced by the formaldehyde fixation. By reversing this cross-linking, we're going to improve the availability of epitopes for tumor cell enrichment. The sample is then processed using the gentleMACS Octo with heaters, followed by filtration through 70-micron filter. Finally, you now have a single-cell suspension that's ready for downstream applications such as tumor cell enrichment. So we have demonstrated that the FFPE tissue dissociation kit preserves 2 very important epitopes for distinguishing tumor and non-tumor cells. This is cytokeratin and vimentin. Cytokeratin is a marker for epithelial cells, and it can also be used as a marker for carcinoma cells. Carcinoma is a type of cancer that develops from epithelial cells. Carcinomas are a very prevalent form of cancer and account for about 80% of all malignant tumors. Carcinomas are often found embedded in mesenchymal tissue, such as the example image of an ovarian carcinoma here. Stromal cells and tumor-infiltrating lymphocytes express the mesenchymal marker vimentin, whereas the carcinoma cells express cytokeratin. And in the merged image here, all the way to the right, you can see the vimentin and cytokeratin staining are mutually exclusive and thus are really good markers for separating out carcinoma cells from non-tumor cells. Now let's look at some data generated by Miltenyi's R&D team demonstrating how well this works and how we can use these markers to enrich for carcinoma cells from FFPE tissue. So looking back at our workflow, we go straight from separating carcinoma cells from non-tumor cells into DNA sequencing. However, given that this is likely a new method for you, you'd probably like to make sure that the dissociation and cell enrichment step worked first. So let's go ahead and add a step to our workflow in order to confirm that our tumor cell enrichment step worked. We used flow cytometry as an extra quality control measure to confirm that we successfully isolated highly pure populations of tumor cells by either using flow sorting or MACS technology. Before we move on to the results, let's review the two options we have validated for enriching for cytokeratin-positive FFPE cells. The first option is cell sorting, which we perform by staining dissociated FFPE cells with cytokeratin and vimentin reaffinity antibodies, followed by sorting on the MaxQuant Pito cell sorter. However, this technique would work with any cell sorter. The other option is to use MACS technology, which is a fast and simple method for separating cell populations by labeling epitopes with specific antibodies that are conjugated to magnetic microbeads. The way this works is first a short incubation step is performed to bind the anti-cytokeratin microbeads to the carcinoma cells. Then the cell suspension is passed through a MACS column that is placed on a magnetic MACS separator. The labeled cytokeratin-positive cells will be held inside the column by the magnetic field, while the non-tumor cells pass through the column. Finally, the column is removed from the separator, and therefore the magnetic field is removed from the column, and the cytokeratin-positive cells are eluted as the enriched positively selected cell fraction. Due to their small size, MACS microbeads do not need to be removed for downstream applications, so you can go straight into flow, sequencing, or whatever else you're doing. So now let's take a look at some data. Here we have an example of human colon carcinoma cells that were purified from a mixture of dissociated FFPE tumor cells that were spiked into dissociated FFPE spleen cells. We enriched this mixture using the cytokeratin microbeads, and the cells were then fluorescently stained with cytokeratin antibody and vimentin antibody and analyzed by flow cytometry. The graph to the left shows the cell mixture before cell separation, and you can see that less than 10% of the cells were positive for cytokeratin. After cell separation with MACS technology, greater than 95% of the cells were cytokeratin positive. We saw similarly good results with cell sorting. In this example, we collaborated with investigators at the Institute of Pathology at the University Medical Center Göttingen in Germany, and we analyzed FFPE pancreas carcinoma tissue. After dissociation of the FFPE tissue using our tissue dissociation kit, we performed cell sorting based on cytokeratin and vimentin expression using those two antibodies. Before sorting, 33% of the cells were cytokeratin positive, while 44% of the cells were vimentin positive. After sorting, the cell populations were enriched to over 95% and with excellent cell recovery, which is shown in the table here. We then extracted DNA from the post-sort populations. From around 200,000 cytokeratin-positive cells, we got almost 2 micrograms of DNA, which is well more than enough material for sequencing and other molecular applications. So before we did do sequencing with these samples, but before we even started that, we went ahead and checked the DNA integrity. Naturally, we were concerned that added tissue processing step involved in the FFPE dissociation process might cause DNA degradation. So we isolated DNA from the same FFPE sample either with or without dissociation. We then ran these samples on a bioanalyzer to examine the quality of the DNA recovered. Notice that the peaks of the red traces appear at the same location and are similar for both DNA isolated directly from the FFPE curl or DNA isolated from the FFPE after dissociation. This indicates that the FFPE dissociation process did not result in any additional shearing or degradation of the DNA sample. So now that we know that our dissociation process did not affect the DNA, we then proceeded on to targeted NGS sequencing using an in-view all-in-one oncopanel. This table shows the results of the frequency of a select number of mutations from targeted DNA sequencing of the pancreatic carcinoma sample. We assessed mutation frequencies for the sample before dissociation, after dissociation, but before sorting, and then we also looked at the 2 sorted populations, the Miltenyi-positive cells and the cytokeratin-positive cells. First, please note that there are no significant differences in the mutation frequencies of the sample either before and after dissociation. You can see this in the blue and magenta bar graphs. This demonstrates that the dissociation process does not affect mutation allele frequencies. We then looked at how separating the cells into tumor and non-tumor population affects these mutation frequencies. 2 mutations, KRAS G12V and TP53R273H, showed a substantial difference in mutation frequency between the cytokeratin and the Miltenyi cells. We expect the cytokeratin cells to represent the tumor population and the Miltenyi cells to represent the normal population, with DNA comparable to the germline. Interestingly, both the KRAS G12V and TP53R273H mutations were virtually undetectable in the Miltenyi cells, but were present in the cytokeratin cells at high frequencies, greater than 50%. This is a strong indication that these 2 mutations are cancer-associated somatic mutations rather than germline mutations. If they were germline mutations, they would not necessarily be associated with the cancer pathology. In this example, both of these 2 mutations were present in the non-dissociated tissue at frequencies greater than 10%. So it is likely that we would have detected these mutations without enriching for cancer cells. However, we just, as you can see, there's a lot of different mutations there. We would have not always been sure whether or not these were germline mutations or somatic mutations without having that matched normal Miltenyi cell control. However, let's look at some examples where the mutation frequencies were not that high. So in that case, not only do we get assurance that this mutation is associated with the cancer pathology, we also get increased sensitivity. In this study, we collaborated with investigators from the Institute of Pathology at the University Hospital Cologne in Germany to examine the effect of enriching for tumor cells in samples with decreasing tumor cell content. In this experiment, we used FFPE tumor samples resected from non-small cell lung carcinoma patients. A trained pathologist estimated the tumor cell content of each of the 3 specimens and circled the tumor cell areas. So specimens 1, 2, and 3 were estimated to have tumor content of 70%, 10%, and less than 5%, respectively. Detection of specific cancer mutations becomes unreliable when tumor cell content is less than 10%. This is because typical NGS assays will not reliably detect mutations when the mutation frequency is much less than 5%. So we took serial sections of each specimen and then enriched for tumor cells either by manual microdissection, that is when we selectively scrape off the tumor from the slide with the scalpel, or by using the FFPE Tissue Dissociation kit in combination with MACS technology to isolate cytokeratin-positive cells. We then extracted the DNA and performed NGS analysis using a targeted oncogene panel. For each specimen, we assessed how macrodissection compared to our method for tumor cell enrichment for detecting cancer mutations. Specimen 1 was largely tumor cells, approximately 70%. And for this sample, macrodissection gave very similar results to our method. In specimen 1, we found a 52% frequency of our driver mutation, whereas with our method, we found a 60% frequency, so not much of a significant difference there. The differences became more apparent in specimen 2 because this sample only had 10% tumor cells. We saw a much greater difference in the frequency of the driver mutation between methods. Macrodissection resulted in an 11% mutation frequency, whereas our method resulted in a much higher 28% frequency. And finally, the most significant difference between the 2 methods became apparent in specimen 3, which contained less than 5% tumor cells. In this sample, macrodissection only resulted in a 1% frequency of our driver mutation, whereas our method resulted in a 7% frequency. Going from a 1% to a 7% mutation frequency is really significant because it's the difference between being able to confidently detect that mutation that is present and not being able to detect that. Most NGS assays cannot reliably call a mutation at 1%, so either this mutation would have been missed or follow-up assays would have been required to confirm it. Whereas if you saw a 7% mutation, you would have definitely detected that with high confidence. So we have focused on dissociating FFPE tissue for NGS sequencing and have demonstrated how tumor cell enrichment can really aid in that sequencing process and identifying of cancer-associated mutations. But sequencing is not the only way you can assess DNA content of a cell. So let's look at another interesting method. Flow cytometry can be used for Ploidy analysis of a cell suspension, and we have found that it works well on dissociated FFPE tissue. Ploidy refers to the number of chromosomes in a cell, and aneuploidy refers to cells with too many or too few chromosomes. Aneuploidy is common in rapidly dividing cancer cells and has been investigated as a prognostic marker in several cancers. In flow cytometry, Ploidy analysis involves staining the cells with a fluorescent dye that binds to the DNA, such as DAPI or PI, and then analyzing the resulting data as a histogram plot, such as the one shown here. In this example, we're looking at normal, healthy diploid cells. Here, the majority of the cells are not actively dividing, so you see a large sharp peak of diploid cells in the G0 or G1 phase. For dividing cells, you see a smaller peak to the right that represents higher DNA content as the cells are duplicating their chromosomes in preparation for mitosis. This same technique can be used to measure the ploidy of a cell population as long as you have a reference diploid cell population for reference. The workflow for Ploidy analysis is similar to what we did for sequencing. First, we dissociate FFPE tissue using the tissue dissociation kit with the gentleMACS octoaceters. Then we sting the cells with fermenting cytokeratin, DAPI, and a red blood cell marker CD235A. Finally, we analyze the samples by flow cytometry on a max quant analyzer. We demonstrated Ploidy analysis with 2 FFPE samples, a prostate carcinoma and a colon carcinoma. The first gating step we performed was to exclude red blood cells from the analysis based on positive CD235A staining. We also gated out non-nucleated cells by the absence of DAPI staining. As I mentioned before, Ploidy analysis requires a reference diploid population. In this example, we're actually able to create our own reference population by gating for the Miltenyi-positive cells and cytokeratin-negative cells. Conversely, we're able to analyze the tumor cells only by gating for cytokeratin-positive cells and the Miltenyi-negative cells. In the resulting DNA histogram, the reference cells are shown as a green trace, whereas the carcinoma cells are shown as the yellow trace. For the prostate sample, the traces of the carcinoma cells and the reference cells are almost identical. This can be quantified as the DNA index, which is simply the ratio of the DNA content of cancer cells to reference cells. For the prostate sample, the DNA index is close to 1, which indicates the prostate carcinoma cells are not aneuploid. For the colon sample, the traces for the carcinoma cells and reference cells are very different. For the colon carcinoma cells, you see two peaks instead of 1. The first peak, peak 1, aligns with the reference peak. This indicates that a portion of the carcinoma cells are diploid. However, the presence of the second peak to the right of peak 1 indicates that a portion of these cells have a higher DNA content than the reference cells and so must be aneuploid. So as you can see, this experiment demonstrates the utility of the FFPE tissue dissociation kit to generate a matched control sample from the same FFPE block when you don't have a matched normal sample available. So that wraps up all the experiments I wanted to show you today. So I would just like to quickly review what we went over in today's webinar. So the most important thing to take home is you can make a single cell suspension out of FFPE tissue. It's not well known, but it's actually very simple to do, and we make it very simple with our FFPE dissociation kit. Importantly, when you are dissociating the FFPE tissue, if you do want to do downstream cell separation, it will be important to make sure that you are preserving your cell markers of interest, and we've demonstrated that we can preserve the cytoskeletal markers of vimentin and cytokeratin to separate out tumor cells or carcinoma cells from non-tumor cells. We have also validated this workflow on the analysis of DNA. So all of this allows you to reduce tumor heterogeneity to get that more sensitive cancer-associated mutation detection. And it also, importantly, removes the need to always have a matched normal tissue control with your tumor samples, which is not available in a lot of FFPE archives. So that's all for today. I'd like to thank everyone for listening, and I'll now open up for questions.
Awesome. Thank you so much, Carina, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we're going to answer as many of your questions as we have time for. Okay, I see a lot of questions coming in. So Carina, our first question that I see is, how do you know that you are fully disassociating the FFPE tissue into a single cell suspension?
So if you're doing this in your lab, one of the easiest things to do is just to look at it under a microscope to confirm this. If you have access to a fluorescent microscope, you could do a DNA dye like DAPI or Hoax to confirm that you only have one nucleus per cell. The way we've done it is by using flow cytometry, and we saw a lot of that data where you can see the clear cell populations that show that this is an individual cell with one copy of the genome.
Thank you. Okay, so our next question coming in is how long an FFPE sample can be stored and under what condition which enable for which, I think they mean which can be enabled for future cell isolation, and will the fixative used influence the results?
Yeah, so that's a great question. I do not have an answer for how long can an FFPE sample be stored, especially if you're getting it from archival samples. You may have FFPE that had been prepared a number of different ways, maybe not all in the same fixative. So the fixative used will influence the results. So we tested everything on formalin-fixed tissue. That is kind of one of those things where you're just going to have to try and see. So when we were validating this protocol, we were not specifically pushing the limits of FFPE age, but you could definitely assume within a few years it should be fine. Oh, I should also mention, it will, just to clarify, it will not work if the section has been used for histology. So if you have an FFPE section on a slide, that will not be compatible with dissociation because those sections are too thin and won't yield any intact cells.
Great, thank you, Carina. The next question I have is, can these first steps be done on a standard robotic setup, or does it need to be manual?
We do not have any solution to do the, we don't provide a solution to do the first few steps, which is deparaffinization and rehydration in a robotic way. However, if you had a solution for that, it could possibly be compatible. We suggest just putting the tissue in the tube, our gentleMACS tube, but if you have a way of transferring the tissue delicately from your robotic system to a gentleMACS tube, then you would be able to do that robotically. But again, this is not something we have a solution for.
Got it, thank you. Our next question, can this be done for mouse tumors as well?
So the dissociation step should work on any number of species and tissue types. However, the downstream separation is only for human cytokeratin. So you would have to then validate your separation on using a different antibody for cell separation.
Great. I would like to thank the audience for all of these questions coming in. Our next one is, why isn't germline mutations associated with cancer pathology unlike somatic mutations?
I have to apologize for that. I think I was a little unclear in my text. So what I meant to say is that germline mutations can certainly be involved in the cancer pathology, but a lot of times when you're looking at tumor tissue as a biomarker for precision medicine, you're much more interested in the somatic mutations, so the mutations that are only present in the cancer cells, because that helps you more clearly define what therapy course to take.
Okay, the next question I have is, can the isolated cells be used for downstream protein extraction?
That is not something we have tested. However, we do know some proteins stay intact. Obviously, we're using cytokeratin and vimentin, but it's just not something we have tested. So you can certainly try.
Awesome, thank you. Let's see, the next one we have is the key step is the enrichment of tumor cells. What is the efficiency, or the efficacy rather, of enrichment of fixed and non-fixed tumor cells with your cytokeratin antibody?
So if we were using non-fixed tumor cells, so I'm assuming by non-fixed you mean fresh live tumor cells, we wouldn't be using cytokeratin. We would need a surface marker. So cytokeratin is part of the cytoskeleton, and you wouldn't be able to access it in live cells. So we have, actually Miltenyi has a lot of great solutions for cell separation based off surface markers of live cells. I highly recommend visiting our website. We use cytokeratin for FFPE just because you do lose a lot of protein signal when you're doing this formalin fixation, then dissociation steps. So we wanted to use something that was like really abundant, and so that's why we use these cytoskeleton proteins. However, it's certainly possible that you could look into other protein markers. Actually, I think that is one of the next questions.
Yes, the next question.
Okay.
Can you look at protein biomarkers in the sorted cells?
Yes. So sorry if that wasn't clear. A lot of that data was for flow cytometry, and we have tested several different epitopes and protein biomarkers in the FFPE tissue. Unfortunately, as I mentioned, you do lose a number of them. So we do recommend doing some optimization and testing before landing on your protein biomarker, especially for surface epitopes. Those tend to be cleaved a lot. So I see one question where they said they're specifically interested in CD3, CD20, and CD138. We have not had a lot of success in getting, in preserving those surface markers for downstream flow cytometry. However, that doesn't mean it's not possible. We did not try everything, and I can definitely follow up with you with some suggestions.
Got it. And have you attempted to do single-cell RNA sequencing from FFPE dissociated cells, or have you tested any optimizations to your FFPE dissociation method to enable single-cell RNA sequencing?
That is not something we have tested. I would, if you were to go down that path, I would recommend possibly optimizing the protocol. We do have that very long 1 hour at 80-degree antigen retrieval step, which is very necessary for cell separation to get access to those protein cell markers. However, you can imagine that's probably not doing that's probably not helping the integrity of the RNA. So you would probably need some optimization of the protocol, specifically the antigen retrieval step. And of course, your starting FFPE sample would need to have decently intact RNA to begin with.
Awesome. And do you have markers for lymphoid elements?
We do not at the moment. We were not able to identify any surface epitopes that worked. Although, again, I would emphasize that we did not try everything. So I don't want to tell you that everything is not possible, but we did not have a lot of success with surface markers.
Got it. The next question I have coming in is, what were the tissue inputs, and in parentheses, core needle biopsies, resections, and tissue quantity that were used for the experiments shown in this presentation?
So in this presentation, we typically had surgical biopsies or resections for this tissue. I think it would definitely work with a needle biopsy. You just might, depending on the area of the tissue, it's possible that you might have to use more than one FFPE section or curl in order to get an adequate number of cells.
Great, and we have time for one or two more questions. The next one is, what is the duration taken for FFPE extraction using this technology?
I think that question is asking how long is the workflow, and it's about roughly 2.5, 3 hour workflow. It kind of depends. So we have the hour-long antigen retrieval steps, and the dissociation step is another 45 minutes, and we have washing steps. So somewhere between 2 to 3 hours total.
Awesome. And our last question for today, using this method, can we isolate cancer cells to find non-somatic cell mutations?
Well, that was our exact idea for using this method, that you could isolate cancer cells out of your FFPE tissue so that you can, so we envision people being more interested in the somatic mutations in those cancer cells, but if you then have normal tissue or normal cells to compare it to, then you can also determine whether or not the mutation in that cancer cell, whether it was germline or somatic.
Awesome. Thank you so much, Carina. Carina, do you have any final comments for our audience?
Just that we have a really great global technical support team. So if you guys, I'm sorry that we don't have time to get to every question today, but please reach out to our technical support team, and you can find all that information on our website. They're really great. They'll get back to you and talk to you specifically about the needs of your projects, and they can definitely answer any specific questions about your sample type. So again, I apologize we can't get to everyone today, but definitely reach out to our technical support team.
Thank you again, Carina, for your time today and your important research. We would like to also thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, stay healthy. Goodbye.